the prolonged intracellular accumulation of. Okay, please.
Well, thank you so much for the invitation to present my work. So the work that I'm going to present is in the context of leukemia. So leukemia is a major health problem. So there are hundreds of thousands of cases of leukemia per year. There are a few groups of leukemia. So we are very interested in acute myeloid leukemia, which represents approximately 37%. Okay, so these are the current treatments for leukemia. So there are a few platforms, therapeutic platforms based on CLINAM combined with cytarabine or retinoic acid with arsenic trioxide. We are quite interested in retinoic acid, so we have developed a few formulations for the release of retinoic acid. So we thought that retinoic acid could be a very interesting therapeutic approach for these. And in fact, it's being used in the clinic. But the issue is there are 25% of patients that once treated with retinoic acid, they have this syndrome. And mostly importantly, in some case, there is the relapse of the patients treated with this drug. So the cells, leukemia-propagated cells that resist to therapy, they still accumulate in this tumor niche. So the retinoic acid, in fact, is transported naturally, physiologically by a protein and then acts in the nuclei. So we thought that it could be very interesting to use nanoparticles to achieve or to reach the nuclei and then to tackle a differentiation program of these cells. So the hypothesis of this work was to develop a system that could very easily release high content, very efficiently, this retinoic acid. And secondly, coming up with the concept that potentially we could use leukemia cells to treat leukemia. So basically based in the tropism of leukemia cells to reach the tumor's niche in the bone marrow, to reach with the cargo and potentially activate by a remote trigger, in this case light, which could start the differentiation of these cells, which by paracrine effect could somehow induce the differentiation of the cells of the tumor in the bone marrow. So this is a different concept from other ones published recently where they use these light activatable nanoparticles to accumulate in the tumor by passive diffusion through these leaky vessels. But essentially based in the leaky of the vessels to accumulate in the tumor. So the formulation is based in a very simple principle where we do a polyplex with an antenna, light-sensitive antenna, in this case the MMC. And so we form a polyplex with a polyanion, and those are the features of these nanoparticles, encapsulating disorder of retinoic acid per milligram of nanoparticles. They are sensitive to UV and blue laser. Depending the power, you have different kinetics. We can activate them inside of the cells. So these are already the nanoparticles inside, labeled with quantum dots. So we can photo disassembly the formulations inside of the cell. But a very important feature of these formulations is the easy how they accumulate in the cytoplasm. So we can accumulate these formulations in two different leukemia cell lines for at least 6 days. These are based in flow cytometry, and these are in CPS results. So based on these, and also considering that these are leukemia cells having PGP, and they could somehow exocytose the formulation, and we demonstrate this with the magnetic nanoparticles. we thought that these could give us a chance somehow to remote activate these cells once we inject in the animals. So they get inside by clathrin and micropinose mediated endocytose by chemical, and we demonstrate this by chemical and genetic inhibition. And after 2 hours, they accumulate immediately in the cytoplasm. So to characterize the efficiency in the differentiation, we did different assays based in the short-term and long-term cell assays, which measured the functionality of the cells for a certain time. And we see that these formulations are more effective than soluble retinoic acid complementing the medium. And once they are activated, they are much more effective than the non-activated formulation, although we have, in this case, some leaching of the retinoic acid from the formulation. We did also, so these assays were done with human leukemia cells from patients. These were with, some leukemia cells, cell lines. So in this case, again, we have the same results, and we develop a reporter cell line which measures the intracellular levels of retinoic acid inside of the cells. And again, after the light activation, we have much more efficacy in terms of, in this case, of activation of the retinoic acid alpha receptor. So in order to demonstrate these, that it's feasible to activate these formulations inside of the animal, so what we did was to transfect leukemia cells with these formulations. And then we developed a specific device for the optical laser activation. So we construct, so they were encapsulated in a gel, and after 24 hours, they were activated. And we see that we can activate the cells after they were subcutaneously transplanted. Well, to demonstrate the concept that it's feasible to do this to reach the bone marrow niche, the leukemia stem cell niche, what we did was to label GFP leukemia cells, inject in the animal to reach the bone marrow. And then what we did was, using the same cell line, we transfect with this light activatable formulation. We inject in the animal, and after 48 hours, 48 hours, we monitor if these GFP labeled THP1, which we call target cells, were in the proximity of the delivery cells. And that's in fact what happened. So showing that these cells, even with the cargo, they can have tropism to the leukemia cell niche. So I hope that I demonstrate that it's possible that these label cells, transfect with the cargo, they can reach the leukemia niche. But then it's necessary to demonstrate that it's feasible to activate these cells once they reach the niche, and afterwards to show that after the differentiation, they can have this paracrine effect. So for that, we design an experiment where we inject these leukemia cells, and we after a few days, when they reach the bone marrow niche, we did some experiments to follow the kinetics of this process. We activate the calvarium, so it's a very thin bone, transparent bone in the mice. So we could use this, in this case, a blue laser for the activation. And once we do that, we wait for more 3 days, and we just in the animals that were photoactivated, so we could see that these cells differentiate into CD11b positive cells, so showing that these cells were differentiated by the cargo that they transport. And also, we showed that once we do that, the cells, the differentiated cells, start to secrete factors that can start the differentiation program of the cells in the vicinity. So in this case, we monitor the process by following the expression of a protein called, it's a transporter, in fact, of the retinoic acid called CRAB-P2. And so these cells in the vicinity, only those cells are activated and start to express this protein. So in order to clarify the mechanism, what we did was to use the reporter cell line that I showed you before to see that once these leukemia cells are activated by the laser, if we extract the supernatant and we expose them to that reporter cell line, if we could see the differentiation. And we clearly see this. So once we activate these cells by the laser, and if we collect the supernatant has these kind of vesicles called exosomes that have the retinoic acid inside, and they diffuse and differentiate the reporter cell line. So again, showing that it's possible to do this and also clarifying the mechanism behind the results that we have in vivo. So I hope to convince you that we have a system that we can use for spatial temporal release of retinoic acid, very small molecule. And we believe the reasons behind the deficiency of this process are connected to this combination of factors. We have data showing these cells transfect with these nanoparticles can be activated after a few days. And although we demonstrate this with a very small molecule, retinoic acid, we believe that we can extend these to other molecules for this purpose. Thank you so much for your attention.
Thank you. Yeah. Questions? Yeah, please.
Thank you. Very interesting talk. I'm a little bit concerned with your use of THP1 cells because they're monocytic. Actually, they are used as a model of monocytic cells, or immortalized that are phagocytic, endocytic, etc. They can be immunogenic because you are repeatedly administering them at least a couple of times to the same animal. Can you comment a little bit on that? Yeah,
so we tried to do. So initially we tried to do with and before, but today the homing efficiency was very, very low. And so clearly what we were tackling was leukemia cells that you could engraft and could initiate this niche in the bone marrow, because it's not easy using other cell lines, at least according to our experience. At least in the AML, I'm not saying the LLL, but the AML was not so easy to have this capacity to engraft using these leukemia cell lines. So THP1, in fact, was one of the few that did this with reasonable success. Yeah.
Okay. Yeah.
So what about the toxicity of orthonitro moieties that you use in your systems? Because when you use light activation, you generate radicals also with this kind of moieties.
Yeah, that's a good point. I didn't show, but we have some in vitro data regarding the effect of the light itself in terms of DNA damage and so on, and of course in the differentiation process. And we didn't see the effect of the light in terms of activating the differentiation program of these cells. So it's and also based in the reported cell line results, we clearly see that the differentiation pattern is connected to the activation of HAR alpha receptor. So connecting the two pieces.
Okay, so in my understanding, your strategy is just kind of a Trojan horse-laden leukemic cells re-injected after loading, and then those leukemic cells are going to find the bone marrow niche. So the question follows, Is there any other light activatable nanoparticle? I mean, you have used the blue and UV laser. So don't you have any infrared? So please imagine the blue laser cannot penetrate the bone marrow of the metaphysis, and it's kind of...
Yeah, so we are developing that right now. So we have already some systems that we are currently exploring for this.
Mm-hmm.
Yeah.
So, and also what might be your strategy, how to load your nanoparticle in clinical situation? Just I imagined.
Yeah.
Draw leukemic cells and then do the chemotherapy and eradicate all those cancer cells. But there might be some other coming up, and then leukemic cells is loaded, as I told you.
No, that's a good question. I mean, this will not be as a first line of therapy. This will be like a second line of therapy. It's just for a few cases that there is, relapse. And so that's part of the reply. The second part is we demonstrate with leukemia cells because of the tropism. So they are much more competent to reach the sites of this leukemic niche. But of course, this concept could be explored with the other type of stem cells, like mesenchymal stem cells or whatsoever, that could reach the niche.
Okay, thank you. Then the next